Honorable guests, respected faculties, participants, and my dear friends, Namaskar, and a very good morning to everyone present here. I, Shubhangi Padi, research scholar from Department of Food Process Engineering, feel privileged to welcome you all on behalf of the organizing committee to the inaugural session of Global Initiative of Academic Networks GIAN course on precision fermentation for sustainable manufacturing of bioactives and industrial biochemicals, sponsored by Ministry of Education, Government of India. I now request our Director, Sir, Professor K. Uma Maheshwar Rao, Dr. Ashutosh Singh, University of Guelph, Canada, Dr. Sabyasachi Mishra, Sir, HOD, Department of Food Process Engineering, NIT Rourkela, Dr. Kunal Pal Singh, Sir, HOD in charge, Department of Biotechnology and Medical Engineering, NIT Rourkela, Local Coordinator, Dr. Sishir Kumar Sahu, Sir, NIT Rourkela, Coordinator, Dr. Winny Routray, Ma'am, and Dr. P. Balasubramanian, Sir, to please occupy the seats to make this day a blessed one. I would like to request the dignitaries on the dais to do the honor by lighting the lamp.
Several different kind of programs, and before also and during this as well, he has helped us to inspired us in many ways so that we can have this program smoothly. And last but not least, our foreign faculty, Mr. Ashutosh Singh, who has graced us from University of Guelph. Professor Ashutosh and me, we were friends. We have been friends since last 15 years. We were in the same lab, and his supervisor is my supervisor's supervisor. So I'm very thankful that he has taken this pain for traveling from Canada for this specific program, and will be with our students and the students from outside who have graced us from around the country. And I hope you all will definitely enjoy this program. And this will be very interactive program, and we hope that you gain the most out of it, all the students. And welcome to the campus, Professor Ashutosh Singh, and all the students who are from outside, and our inside students, without whose participation this program will not be possible. Thank you very much.
Thank you, ma'am. Now, I would like to request Dr. Sabyasachi Mishra, Sir, Head of the Department of Food Process Engineering, NIT Rourkela, to please address the gathering. Please, sir.
Good morning, everyone. Dignitaries on the dais, and faculty members, colleagues, students, and participants from across India. I got to know that for this particular GIAN course, 32 students from across different parts of the country, they have registered, and few are on travel because of this cyclone and the weather. Trains are delayed, so probably they will be reaching in an hour or two. So with this particular GIAN course, probably this is the 8th GIAN course from our department, sir, in a short span of time. And being the head of the department, I take it my pride. We have 16 faculty members at present. We are full. And then we have currently 46 PhD students working. And in last 10 years, we started the department in 2013-14. In last 10 years, we have published 786 SCI publications. We secured 64 sponsored projects. And many of our students, they are working outside, campus placement going on. Last year it was 91% placement, and in the resource optimization parameters also we stand second in the research, sir. So we have been working really hard, and for all these things our administration has supported us in a big way. And recently also I must mention that we have some space issues. So I requested Professor Rao, and Rohan sir is also there. And very promptly we are going to get few spaces, few rooms. And in addition, in principle, Director sir has also agreed for a separate building also in next 1-2 years for the department. And hopefully hopefully we'll get those things. Coming back to GIAN course, this is based on a fermentation technology. It has a wide industrial application, starting from alcohol and alcoholic beverage, non-alcoholic beverages, probiotics, which I work on. So it has so much of like applications in industrial scale, and I hope during this particular GIAN course, all the participants and our students who are there, they will learn a lot from Dr. Ashutosh. I really thank him. I have a deep connection with Canada, and just today morning I got to know that he went to University of Saskatchewan for some work under my supervisor also. So that connection is also there. So thank you for accepting our invitation and be part of this GIAN course, and I hope our students will learn a lot from you and will have deeper collaboration with you in future. And thank you, Sisir sir, for taking all the pain, coordinating so many. I think this month, sir, we have another 2 more GIAN course from the department. Yes. And my colleague, Professor Kunal Pal, in charge HOD, thank you. And we have been partnering. I think after this inauguration is over, we'll again go to BM department. That is also a food processing and BM department collaboration Spark project. And I hope your stay and your learning in this campus will be really pleasurable. And thank you.
Thank you, sir, for giving such a beautiful insight to our department. Now I request Dr. Kunal Pal, sir, HOD in charge, Department of Biotechnology and Medical Engineering, NIT Rourkela, to address the gathering. Please, sir.
Good morning to all the dignitaries, participants, colleagues, students, and esteemed guests. It gives me immense pleasure to welcome you all in the inaugural session of the GIAN course on precision fermentation for sustainable manufacturing of bioactives and industrial biochemicals, organized jointly by the Department of Biotechnology and Medical Engineering and the Department of Food Process Engineering. We are honored to have with us today our Honorable Director, Professor K. Umaheshwar Rao. Sir, your esteemed commitment to fostering a vibrant academic culture to our institution serves as a constant inspiration. Your support has been pivotal in enabling us to host such knowledgeable sharing initiative. I would also like to extend a welcome to our registrar, Professor Rohan Dhiman, and Professor S.K. Sahu, local coordinator, whose efforts contribute significantly to the smooth functioning and academic excellence of our institute. Our heartfelt thanks and welcome to Dr. Ashutosh Singh, Associate Professor at the School of Engineering, University of Guelph, and the key speaker for this course. Dr. Singh's expertise in the field of precision fermentation and sustainable manufacturing will undoubtedly enrich our understanding and open new avenues for research and development in this critical domain. As the head in charge of the Department of Biotechnology and Medical Engineering, let me take this opportunity to introduce our department to this distinguished gathering. Established in August 2007, the department currently comprises 24 faculty members, all holding doctoral degrees from reputed institutions. Our faculty are actively engaged in cutting-edge research and have garnered financial support from prestigious agencies such as DST, DBT, CSIR, ICMR, and SERB, among others. These grants have significantly contributed to the enhancement of our research infrastructure. The department houses state-of-the-art laboratories, including the Medical Electronics and Instrumentation Laboratory, Cell and Molecular Engineering Laboratory, Gene Manipulation Laboratory, Environmental Engineering Laboratory, Rehabilitation and Regenerative Medicine Laboratory, Biomaterials and Tissue Engineering Laboratory, Smart Materials Laboratory, and many others. These laboratories serve as the hubs of innovation, providing our students with hands-on training and equipping them for industry-ready challenges. This workshop, under the aegis of Global Initiative of Academic Networks, aims to shed light on the transformative potential of precision fermentation technologies in sustainable manufacturing processes. Over the course of program, we will delve into the nuances of advanced fermentation techniques and explore their application in creating bioactives and industrial biochemicals. Before concluding, I extend my heartfelt gratitude to the coordinators, Dr. Balasubramanian and Dr. Winny Rourkela, for their dedication and meticulous planning, ensuring the seamless organization of this course. My thanks also go to the participants whose enthusiasm and active engagement will undoubtedly make this event a success. Once again, I welcome you all to the exciting journey of learning and collaboration. Let us use this platform to foster knowledge exchange and inspire innovation solutions for a sustainable future. Thank you. Jai Bharat.
Thank you, sir. Now, I request Dr. Sushir Kumar Sahu, sir, local coordinator of GIAN, NIT Rourkela, to please address the gathering. Please, sir.
A very good morning to all. For the title of the course, Precision Fermentation for Sustainable Manufacturing of Bioactives and Industrial Biochemicals, respected director, NIT Rourkela, Professor K. Uma Maheshwar Rao, the foreign expert of the course, Dr. Ashutosh Singh, University of Guelph, Canada, the registrar, Professor Rohan Deman, Head of Department of Food Process Engineering, Dr. Sabyasachi Misra, and Head of Biotechnology Medical Engineering, Dr. Kunal Pal, Course Coordinator, Winny Routray, and Dr. Balasubramanian. Dear faculty colleagues, participants, students, ladies and gentlemen, as the Institute Coordinator of the GIAN program, which is basically a program of the Ministry of Education, Government of India, designed to tap the talent pool of scientists and entrepreneurs internationally to encourage their engagement with the Institute of Higher Education in India to augment the country's existing academic resources, accelerate the pace of quality reform, and elevate India's scientific and technological capacity to global excellence. GIAN enables institutes of higher education in India to invite and host internationally renowned academicians and researchers, facilitating the co-teaching of advanced courses to Indian students and fostering collaborative relationships with experts from around the world. GIAN offers international experts a unique opportunity to familiarize themselves with India's academic environment by interacting with local students and faculty, which can explore potential collaborations, share their expertise, and contribute to the advancement of knowledge across diverse fields. Coming to the subject, fermentation means, as we all know, means using microorganisms like yeast, bacteria, or fungi to produce specific proteins, fats, and compounds. Day-to-day use, we use breads, cake, cheese, yogurt. So this precise fermentation means actually involves programming these microorganisms through genetic engineering to produce desired substances like animal-free dairy products, egg-free proteins without chickens. So everything is for sustainability. Bioactives are naturally occurring compounds that have biological effects in living organisms found in plants and animals, used in food, pharmaceutical, cosmetics for functional benefits. It may be plant-based such as berries, citrus, fruits, green tea, which have antioxidant and anti-inflammatory properties. Tea, coffee stimulates the nervous system. Or it may be microbial bioactives produced by microorganisms for probiotics and antibiotics. Similarly, industrial bioactives used in packaging, medical implants, cosmetics, and many more. So the GIAN scheme launched in 2015 and 3 phases are completed. The 4th phase is now running under the IIT Hyderabad as the central nodal agency. NIT Rourkela has conducted 24 courses in 3 phases, and the current phase, this is the 3rd course, and ready to conduct 10 more courses in the phase. All best wishes for the persons involved in making the course a success. Thank you very much.
Thank you, sir, for giving us an insight on about the GIAN course. Now, I would like to request Dr. Ashutosh Singh, sir, Associate Professor, School of Engineering, University of Guelph, Canada, to address the gathering. Please, sir.
Thank you. Good morning, everyone. It felt like a class right now. So you all stand up and then say good morning, teacher, or good morning, sir. You don't have to. Welcome to the session, or I'll say welcome to the GIAN program today. It's really an amazing opportunity. Thank you so much. Oh. As you can see, I'm not very used to this, speaking in the mic. I'm way much more a person who would be walking around talking to the students. It's an amazing opportunity. As Dr. Winny said, me and Dr. Winny have been friends for almost 15 years. We did our PhD together, and then she moved into different directions. I moved into different directions, but the world is very, very small, actually. So in this small world, you will be going through a lot of things that you learn. You're going to learn a lot of things here as part of your undergrad or master's or PhD, and you're also going to learn a lot of things as part of this GIAN session. The thing that I would like to definitely mention, and Dr. Sahu was able to actually give you what I'm going to teach. He has already explained that. So I'm not going to go through what fermentation is, what are the different compounds that you can make, but I can definitely state that some of the topics that we will discuss, it won't be something that I'm teaching you, it would be more of an interaction between me and you. Some of the topics that we will discuss, like cellular agriculture or precision fermentations, they are very, very, very current. They are very, very, very important. And if you are looking for a future, let's say in pharmacology or in pharmaceuticals industry, or you're looking in the food industry, this topic is key for you to get into a job prospect that you're looking for. So I will say it would be nice if you will be able to sit during the presentation with an open mind. Because there would be topics that might question your ethical senses. If once we start talking about cultivated meat, it might be something that you are not interested, or you might find very disgusting, let's say. But you have to go through it because that is something that is going on throughout the world. It's not, if it's not happening in India, it doesn't mean that it's not happening anywhere else. So we have to talk about that. And there will be other different topics. We'll be going through a lot of information about different types of fermentation systems or reactors, how you design those reactors, how to apply. And the most interesting part of the course would be the 2 case studies that you would be participating in. And you'll be working as groups of students coming up with a solution. You'll be designing the whole process of developing a new product. So it would be interesting. And I would like to thank everyone who is on the dais, including the director, Dr. Rao, Dr. Kunal, Dr. Sabyasachi, Dr. Balasubramanian, Dr. Sahu, Dr. Winny, for giving me the opportunity to come and talk to you. And I hope you have a very good time with me, and I hope to have a very good time with you. Thank you.
Thank you, sir. Now, I would like to request our director, sir, Professor K. Umamaheshwar Rao, NIT Rourkela, to address the gathering. Please, sir.
Thank you. And the most distinguished dignitaries on dais, the foreign speaker or the guest speaker or the important speaker, Professor Ashutosh Singh from University of Guelph, Canada, and head of the department food processing, Professor Sabyasachi Mishra, and head of the department in charge, right, Professor Kunal Pal, and Professor Winny Routray Madam, coordinator of this GIAN program, and Professor Balasubramanian, co-coordinator of this program, and other faculty colleagues of the food processing and biomedical department, and the participants from the institution and across the institution. A very good morning to all of you, and it gives me immense pleasure to be present here today this morning for this one-day GIAN program. Pardon? 5 days. 5-day program. So 5-day GIAN program on the precision fermentation for the sustainable manufacturing of bioactives and industrial biochemicals. Right. And at the outset, before we really know little, acquire some GIAN on this particular fermentation process, let me just take this opportunity to congratulate both the departments who are together, food processing, for the exceptional performance with the 16 faculty members. Yes, I have promised that I think food processing department needs a space and everything in that space for the food processing. We are trying. One thing is that it is my promise, and Professor Savysachi Mishra knows that certain times when we make a promise, it's not a political statement. Let me tell you very clearly. It is a statement from the bottom of my heart. It is not just about food processing. I am planning to really have some other departments who really are at present sharing this space with others. But then probably what really happens, most of you may know or may not know, that when you are sharing the department with some other department, there may be some induced energy and with which you are producing many magical things. But when you are detached from that one, and then when you are isolated from the department from whom you are sharing, probably, I don't know, right, that should not be deterring, and then in fermenting the thoughts and then trying to manufacture the better results from your department. Right, the topic is something which is right, generally I find at ease in trying to address the people, but then not a single word is really keeping me in comfort zone. Right, all that I know is there are only 2 words which are really absolutely clear in my mind. One is about the precision, the other is about the sustainable thing, of course sustainable manufacturing, okay, this one phrase, but the rest other things are not really, because fermentation is one thing which I know. I am a hardcore and then pure vegetarian, and all that I know is that fermentation is a process for making curd. Fermentation is a process which we have been doing for generations, for fermenting the dough for idli and then dosa and various other things. But then Professor Savysachi said that lot many alcoholic beverages are there, which I really do not know whether there is a need to ferment them or not, right? Because already people are getting high without even those fermented liquids. We are already high, okay? And then I just keep my fingers crossed about of course there is a lot of revenue in that one, all right? So now it only speaks that left, right and center people are really looking for an occasion to put lot of fermented material in their minds and then bodies, and then go little out of control, okay? Just recently I read in paper that on Diwali, one day, Lakshmi Puja day, in Hyderabad city alone, 30,000 crores was the profit, right, incurred by this fermented beverages getting into the stomachs of the people, and therefore, right. But then I really caution that my students that you don't have to really lose yourself, right, out of the context, because we are sitting in a GIAN program. GIAN, I think you all know that it's a Sanskrit word. It's an acquiring wisdom, right? If I really try to say that, you are trying to acquire the wisdom that what is Professor Ashutosh Singh will be deliberating in these 3 days or 5 days, sorry. And I wish all the participants very best, and right, thank you for giving me this opportunity. And lastly, as far as those who are not from NIT Rourkela and those who have come to their first visit to Rourkela or any, it's a beautiful time. It's really a beautiful time. For some reasons that there is cloud spread down the sky, otherwise it's enough chill is there, not like Canada. For Canadians it is a summer, I know that they are accustomed to minus 40, and then because they are very close to the very peak of the globe. And incidentally, all of them are, some of them are non-resident Canadians, some of them are already Canadian citizens. And then I am really very comfortable with the word Canada. The reason is that very often I go to Canada, right, to meet my family, means my son is there. Right, and so again, lastly, I congratulate food processing department for their exceptional work. Okay, and then whatever the help that is required from the administration for anything, it's always there. Wish you all the very success. Thank you very much.
Thank you, sir, for your wonderful message. Now, I would like to request our director, sir, to present the memento to our guest, Dr. Ashutosh Singh, sir.
Thank you for the most one thing which I really like as far as the token of memory is this one. This is the Trinity, and all the folks and dogs are there. This is Lord Jagannath. Okay.
मैं जिसका नाम लूँगी अभी तुम दोगे, मैंने बताया था ना सीसी सर को दोगे, डायरेक्टर सर को देने के लिए। नहीं साथ में एक साथ में लेके जाओ। हाँ साथ में लेके सीसी सर एक ले लो।
The value of this one is about the time which they have spent and then the dedication with which they have done this. And it is not a fermentation process, but there is some precision chemical process of trying to give this palm leaves permanence, to be sustainable for others. Thank you, Winny Dr. Madam, for really giving me one opportunity to honor our guest today with a memorable token of gift. Thanks.
Thank you, sir. I now request Dr. Sushi sir to present the memento to our director, sir, Professor K. Uma Maheshwar Rao. I request Dr. Sabyasachi Mishra sir to present the memento to Dr. Sishir Kumar Sahu sir, the local coordinator of GIAN. I now request Dr. Winny Routray ma'am to present the memento to HOD FP department, Dr. Sabyasachi Mishra sir. I now request Dr. Bala sir to present the memento to HOD BM department, Dr. Kunal Pal Singh. I now request Dr. Sabyasachi Mishra sir to present the memento to a coordinator of the GIAN course, Dr. Winny Routray ma'am. I would now request Dr. Kunal sir to please present the memento to our coordinator of GIAN program, Dr. Balasubramanian sir. Thank you, everyone. Dr. Ashutosh Singh has got some gifts for the dignitaries on the dais as a token of appreciation. So I request him to present them.
cannot say this has been handmade or but it does represent Canadian coffee and culture. If I may, just here for a second. Yes. It has been an amazing opportunity for me to meet actually a lot of people here. So thank you so much for the gift. And this gift is, as the head, sorry, you call it HODs here, so we call it area heads in the School of Engineering because we don't have departments. So as an area head for biological and biomedical engineering, this is a token of gift from my program, my department, our school to you all. And thank you for the opportunity. And I hope this will initiate a relationship between University of Guelph and NIT Rourkela. And we can have a bit more of what we have right now. Thank you.
Thank you, sir. Now we have come to the end of the inaugural session. So I request Dr. Balasubramanian, sir, coordinator of this course, to please propose the vote of thanks.
It's my profound privilege and great joy to extend the vote of thanks to all those who are contributed in one way or other way to make this GIAN course a reality. I thank especially the course instructor, Professor Ashutosh Singh, Associate Professor from the School of Engineering at University of Guelph, Canada, for his acceptance to offer this course through invaluable lectures and hands-on training practical sections, which are well aligned with the GIAN course expected outcomes of gaining invaluable insights, practical knowledge, and international perspective that could enrich the participants of academic pursuits and future careers. We are grateful to the director of our NIT Rourkela, Professor Uma Maheshwar Rao, and our local institute GIAN coordinator, Professor Shishir Kumar Shahu, for their mentorship and support for hosting this GIAN course. We are also thankful to Professor Swadesh Kumar Pradihar, the staff of administration strict, Professor Rohan Diman, Register Office, for providing us with all the infrastructure and logistics support towards organizing this GIAN course at NIT Rourkela. We acknowledge the unwavering support received from both heads of the department, Food Process Engineering and Biotechnology and Medical Engineering, the faculty and staff members to conduct this course. We are very much indebted to Ministry of Education, Government of India, New Delhi, for providing the necessary financial support for conducting this GIAN course. We extend our sincere thanks to our central nodal agency, IIT Hyderabad, who has been the driving force behind the whole process. Their vision and interest has once again culminated in making the course a grand success. We are gratified to all the participants for being so enthusiastic and coming all the way from different parts to NIT Rourkela for attending this GIAN program. We have been fortunate enough to be surrounded by the enthusiastic students from both on campus as well as from off campus. I hope the GIAN course meets the expectation of each and every person present over here. This course will equip participants with the knowledge and skills to leverage fermentation technologies for sustainable manufacturing of bioactive and industrial biochemicals. Let us ensure that the knowledge exchanged during these 5 days drive forward quality innovation in precision fermentation technology, ultimately creating solutions that can improve our lives and shape a more sustainable future and healthier future for all of us. Finally, before concluding, I would like to thank to all our student coordinators who have been working extremely hard from both background and foreground in making this GIAN course a grand success. I hope you enjoy this 5-day long GIAN course and have a nice day at NIT Rourkela. Let's have a stimulating teaching learning exercise together. Thank you.
Thank you so much, everyone, for joining the inaugural session. We have a high tea arranged outside the room. Please join us for that. After 15 minutes, we'll start with the first keynote session.
Good morning. I like the way you all say that, good morning. Anyways, welcome to the first session. My apologies that we have been delayed by around 45 minutes. I hope we could have started earlier. This particular session has been divided into two different topics. So we are going to discuss about introduction to precision fermentation and cellular agriculture. So I'm going to talk about cellular agriculture first, and then I'm going to talk about precision fermentation. So once I will finish the presentation for cellular agriculture, we'll take a break for 15 minutes, and then we'll start the precision fermentation. So before I start this presentation, do you have any questions with respect to the formatting? If you want me to change something, or if you have any questions specific to the topic, or if you want me to talk about something in the topic specifically? Nothing? I was expecting that answer, so thank you. Okay, so very well. Cellular agriculture, and you all have already been informed about my name, or I have been introduced you to you. So my name is Ashutosh Singh. I am an associate professor at University of Guelph. My research is in food and bioprocessing mostly, specifically food waste valorization. Cellular agriculture or precision fermentation is one aspect of research that I am involved in, and that's the reason why I like this topic a lot, and it is something which is very, very current. But before we start getting deep into the core of cellular agriculture, we'll discuss a few things on what would be the outline of the presentation for today. Okay. So I'll give you a brief background on why we are talking about cellular agriculture, why it is an important topic. I will also explain you in the context of alternative proteins and how it is related to cellular agriculture. We'll also have an introduction to cellular agriculture. Now, cellular agriculture is an agriculture, but then there is two types. One is a cultured meat, another one is acellular agriculture, which we basically are aware of most of the time. So we'll discuss about that. And then we'll discuss about the cellular agriculture products. I will give you some of the examples here, mostly about different companies and different products that have been launched in the market right now in North America, in Europe. I don't have many examples from India, but you should be able to connect it in one way or other. And then I will talk to you about the market landscape of cellular agriculture and precision fermentation. And then we'll talk about the challenges, the needs and the opportunities, and this will be the most interesting part, because this is where, as a researcher, for me the opportunities lies. Like whenever I see a specific field of interest, I always try to find what the challenges are there so that I can find a technical solution. As an engineer, that is what we do, we find technical solutions, right? Very well. So food security and population, that's the very first thing that we are going to talk about. So food security, the word security, if I have to call myself food secured, it means that I should have access to enough food, not just any food, but it has to be a nutritious food, good for my health, and it should be affordable enough for me to buy it, right? But what you will find in the current time, after the COVID-19, I have seen majority of the countries in the world, they have inflation. There's a lot of price increase for the common food products that people used to buy earlier. And you will see as an example on the very left-hand side, this is from the Canadian perspective. So if you look at from the Canadian perspective, this is for a family of 4. So I have a family of 4, so me, my wife, my 2 kids. For me, if I have to feed my family well, in terms of like giving them a nutritious food, I have to spend approximately $1,244 a month just on food. And if you're also paying for your house, now that we call as mortgage, or here we call it as EMI, that you pay for your loan. So if I have taken a loan, I pay a mortgage to the bank. So if I'm paying the mortgage to the bank, I'm paying for the car, I'm paying for my kids, let's say medical bills or anything, everything included, I'm left with minus $230 every month. So I don't save any money. So people are always interested in saving, right? Where exactly will you cut your expenses? The very first thing that we cut is on food. We'll start trying to buy alternatives, right? So even if you are earning enough with inflation and the changes that is happening globally, we are still food insecure. We are not food secured. So we always need to find alternatives that are nutritious, but they are also affordable and sustainable. Sustainability is a very key point here. And then if you look at the graph at the very bottom on the left hand side, you will see the world population, how it is increasing from 2019 to 2022. So from 7.9%, it has went up to 9.2%, and how much of that population is undernourished. That undernourished is approximately 735 million. That's even like 2 times or 3 times of the US population. So those many people are undernourished. They don't get enough food or nourished food or good food or healthy food for themselves. The other point is that if your population of the world is increasing, what is the best thing that you can do? You have to produce more. And to produce more, you have to use your land resources. Do we have limited or we have unlimited land resources? It's very limited, right? So if we keep on changing our forest areas into agreeable lands, what will happen is all the agreeable lands will start being diminishing down, and we will have deforestation, right? So we don't want to be in that type of situation. We want to be in a situation in which we are able to produce more with whatever capabilities that we have right now. We don't want to use more. And that is where this whole thing comes up. So what you will see is that with the 56% more food that would be required in 2050 to feed the 10 billion population that we might have in 2050, depending upon what the growth rate is right now, we don't have enough opportunities with respect to the land resources that we have. So we have to find alternative way of producing more food. And that is where this whole quotation from World Resource Institute comes in to picture, or like you will understand, is that to adequately feed more than 9 billion people by 2050, the world must close a 70% gap between the amount of food produced today and that would be needed in mid-century. That's a pretty huge number. Don't you agree? So this is the context that will set up why we are talking about cellular agriculture and why we should be looking into it. Now let's look into the protein consumption part. Now this is a very interesting topic. Now from the Indian perspective, that's why I asked you to have a very open mind. So from an Indian perspective, we are more inclined towards vegetarian protein, the pulses, the cereals, the grains. Those are our primary sources. The milk would be a primary source of our protein. If you are a non-vegetarian or ovo-lacto-vegetarian, you will be going into the egg proteins. That would be one of the sources of the protein. But for the rest of the world, they are meat eaters, right? So their primary source of protein is meat. And that creates a completely different set of problems, because if you see the trends, like for example, India is looking at only 14 kgs per capita meat consumption in 2021. That's a pretty less number. And if you're looking at Portugal, or if you're looking at United States, you're looking at 149 kgs of meat. And in that, primary would be your beef protein, you'll have your pork, and you'll have your other meats. That would be your seafood. Another thing, the 58 kg is basically your poultry. So chicken is the primary source, KFCs, McDonald's, those are our primary sources of protein for most of the kids. But beef, pork, these are also a major chunk of our protein sources in North America. But for India, you will have those as a very small source. Our reliance on grains, cereals is very heavy, right? So that's the reason why our protein consumption from that sources are very less. And as the population increases, do you think the protein consumption will increase? Yes or no? Why? Why do you think that protein consumption will increase as the population increases? So if you have your population and you have your protein consumption, how are they related? If they are directly related, there has to be some sense, right? Then our population is pretty huge in India. Why is our protein consumption not increasing? You are right, yes, we do have alternative sources. Even soybean is available in North America. Why are people not interested in that? Think about this way. So if let's say if you have one person here, so and you have the same person in a different alternative world, here that particular person is, I will go in rupees, is earning around 40,000 per month, and here he is earning 400,000 per month. Do you think the choices of food will change if they are earning more? If you earn more, will you be more interested in buying meat? No, until unless you are pure vegetarian, that would be a different case. But I'm just saying, if you are a non-vegetarian and you have an option, you are earning a lot of money, will you be ending up buying more meat? Majority of the times, that's the relationship that has been seen. So because as the income increases, our reliance on meat protein increases, and that is the reason why you will see this growth difference between India and different other countries, because when you're looking at the per capita income of Indians and the per capita income of other countries, individuals in other countries, our choices of food will differ. It also depends on the availability, what we have, but it also depends upon what we can afford. If I can afford mutton, I will definitely buy mutton, because good for health, right? That's the way we look at it, we perceive it. It's also based on the perception that you have. So that's how the relationship is, because as the population will increase, as your economic value or your own economics increase, you will tend to purchase more meat. Now, meat consumption is related in many ways. So you will have this in this particular graph also from Goodfriend et al. You will see the meat consumption in basically in India has remained more or less constant. For the rest of the country, specifically China, it has increased significantly, right? So it is going like in an exponential curve as the population has increased, as their purchasing power has increased. When was Chinese economy started? When did the Chinese economy started going up? In the late 1990s, right? So if you look at the late 1990s and the 2010, it's an exponential growth. So because their economic progress improved, the income of the people improved, they were able to purchase better food. And if the meat was their primary source of protein, so they were able to purchase more of that. Now, another aspect, now we have discussed about the meat, right? Beef, pork, chicken, they all require land. So land resources have to be used. Now, if you are in North America or in Europe, if you have livestock farming, now when you define agriculture, for example, so agriculture is not just crops, it also has livestock, aquaculture, right, fisheries, and any other? Majority would be these. So you'll have your fisheries, you'll have your aquaculture will be there, You have your livestock there, and then you have the crop. Now, crop is our primary source of food. Then comes the livestock. Now, for the livestock, if somebody has a livestock farm, if let's say somebody has a dairy farm, or somebody has like a beef farm, so where they are rearing cows for meat, or even if they are rearing goats, or if they are rearing, let's say, pigs for meat, they have to have pasture land, they have to provide the feed. That whole thing adds to a lot of stress on the environment. So, more and more land has to be used if you are more focused on the meat production. Might not be in India, but the rest of the world, yes, it will happen. So, the croplands and pastures right now occupy half of all the vegetated lands today. And the environmental degradation of that land use is another aspect that adds to the negative notion to livestock production, because the amount of water that would be required or amount of GHG that is being emitted out is pretty significant. It's very, very high. If you're looking at like from the dietary perspective, if you're in the vegan, you're looking at 1.5 tons CO2 per person. But if you're more of a meat lover, you're looking at 3.3 tons CO2 emitted per person. So that's a huge number in emission of CO2, and that will lead to global warming, right? And that's why even in the COP29, the COP20, in the previous ones, they have always talked about livestock production, how we should reduce or our reliance on the livestock for the meat production. And this particular graph on the left hand side, you will see, so land use, fertilizers, transportations, feed processing, generation of the feed, transportation of it, and then you are looking at the growth of your livestock. All of that, this 39% is just coming from the livestock. The amount of GHGs that are coming out is just from the livestock. That's a huge amount, and that is also another reason why we are interested in looking for alternative to meat proteins. We are looking at plant-based proteins just because of that, because we want to reduce the amount of GHG that are being emitted in the environment. Now, why should we be interested in cellular agriculture? We have already discussed that, and these are the key points from there. So, the animal agriculture is one of the leading causes of global deforestation, because more and more land would be required for the pastures. It is also the major contributor to foodborne illnesses and zoonotic disease outbreaks. If you are not careful, you would have heard about swine flu, avian flu, right? So, those are all related to zoonotic disease, right? So that's what happens if you are not careful. And then it is also leading driver of antibiotic resistance. Now, why? Because when you are looking at livestock agriculture, you want your cows, you want your pigs to be in good health. So, you are basically using a lot of antibiotics to keep them healthy. Now, those antibiotics are there in their body. If the people are consuming it, that leads to antibiotic resistance. So, if it is also there, like if you are pumping antibiotics into dairy cows, antibiotics are there in the milk, that milk you drink, it adds to that antibiotic resistance. So, that's why it becomes the whole chain of antibiotic movement adds to this. We do not want that. So, that is why another aspect would be, because we do not want antibiotic resistance to increase, we should reduce our reliance on the meat, and we need to find an alternative to it. And also the inhumane processing and practices. Yes? Accumulation of antibiotics leads to resistance because more and more you get used to it, right? So, the bacteria, if they are being subjected to the same antibiotics again and again, they are also going through evolution. So, they are also going to have strains that would be resistant to those antibiotics then, and we might not be able to fight them after that. I will give you a very basic example on that aspect. So, when I was in India, if anything happens to me, if I go to a doctor, the very first thing they used to give me is antibiotics. So, when my kids in Canada, if I take them, if they have cough or fever, if I take them to the doctor, they won't prescribe antibiotics at all. They will ask us to like wait for 3 days. If it doesn't subside in the next 3 or 4 days, only then they will prescribe the antibiotics, because they are very careful about this antibiotic resistance development. I am actually immune to many of those antibiotics, don't even work for me. They have to look for higher dosage of that. So that happens. That is actually the case, and that's the reason why we are very interested in this aspect. So it also accounts for 14 to 18% of the global house gas emissions. And you can actually see the antibiotic use for in milligrams per kg. The maximum is being used in Cyprus. I think India was not there in that particular data plan, but you will see US is somewhere there, United Kingdom is there. Yeah, so US is up here, and then UK is down here. Norway is one of the most beautiful country in the world, and they are very, very careful about what goes in their body. And it's also called as the happiest place on earth or something like that. It's also in one of those lists. That is one of the reasons, because they are very careful about what goes in their body, what type of food you eat. You will see, you have heard about the term GMOs, right? Genetically modified organisms. So now GMOs, if I ask, would you like to eat anything which is GMO, what would be the first answer you will say? No, right? No, genetically modified organism is not good for us. But honestly, what percentage of the food that you eat every day is actually GMO? Can you guess? Is it like 10%, 20%, 50%, more than 50%? It's more than 50%. The tomatoes that you buy in the market, genetically modified. Potatoes that you buy in the market, genetically modified. Majority of the food, until unless they are grown organically, they have been using, let's say, the seeds which were natural, wild varieties. That's real. But wild varieties are not there anymore, right? So we are eating a lot of genetically modified organisms anyway. Now, what is cellular and acellular agriculture? Now, the topic is very, very interesting. So, if you look at agriculture, now agriculture is with development of food products. Now, cellular agriculture is also defined as manufacturing of agriculture products like keratin, silk. Now, the silk that I am talking here would be the spider silk. You are using the protein that is the spider silk protein. I can use that, put it into an E. coli, ferment it, and get that silk protein. So, cellular agriculture is defined as a manufacturing of agricultural or animal or bio-inspired, bio-based products from cell cultures. Now, these cell cultures, either they are producing the product or they are the product themselves. So, either you eat the cells that you are fermenting, the growing, those are your product, or the cells are producing a product extracellularly. So, the cell is releasing the product into the cytoplasm, and we can do the cell lysis and extract the product out. So, mostly the proteins that we get. You will get an example in the session tomorrow about insulin. That's one of the extracellular product. Proteins, enzymes that are being used, developed by fermentation process, is an extracellular product. Now, if the cell itself is a product, how do we actually manufacture that? That is a very interesting part. Now, that is where cultivated meat comes into picture. If the cell becomes a product itself, that's cultivated meat is an example, but many a times we just use as a biomass, the whole cell as a biomass or a source of protein for ourselves. Now, if that particular protein was not released, let's say outside the cell, but if it was only released within the cell, I can eat the cell directly to get the whole as a protein source, right? So, that becomes like a cell or cell biomass as a product. Now, these cells as a product itself, for that you have to do the extraction and isolation of stem cells. Now, this is specifically for cultivated meats. Now, the stem cells have the capability of growing into different forms of tissues. So, they can develop fat, they can develop muscles, they can develop skeletal tissues. So, the stem cells have the DNA in them to grow into different forms, and that will, and if you give it a shape, it can give you the whole product. Then you are providing them the cell growth, the proliferation and the differentiations in a bioreactor, and then you harvest it, and that becomes the whole product itself. And then you have the acellular agriculture. Now, the acellular agriculture is something that you should be more familiar with. It includes products obtained or purified from host organisms such as bacteria, yeast, microalgae, which are grown via fermentation. Now, acellular products, as you can see from the left hand side, are mostly genetically modified. So, what you do is you modify the genome or the DNA of an E. coli, you add your foreign DNA into it, or the interest, the DNA of interest, which will develop a specific protein of interest or the product of interest. That is called recombinant DNA technology, right? Precision fermentation, before it became precision fermentation, it used to be called recombinant DNA technology production or processing. It just got a fancy name and became precision fermentation because it's very precise. We are only interested in one specific product, and that's the reason why it is called precision fermentation. Now, it uses techniques such as molecular biology, acellular production will use techniques such as molecular biology, synthetic biology. It is used to produce molecules such as proteins and fats that can be an animal product, milk and egg, or it can be a plant-based product such as vanillin. Now, vanillin is vanilla essence. So, that is another product which is made. Anthocyanin, anthocyanin is a natural color which is found in fruits. And then, since the cells used to produce are not the part of the final product, they are called acellular. If the cell was the final product, it would become cellular or cell based. Now, methodology used to produce these acellular product is referred as precision fermentation, and that is the reason why I wanted to go through cellular agriculture first and then into precision fermentation. So, you can get an idea of what we are dealing with in precision fermentation. Now, fermentation in itself is a very important technology. As a unit operation, it has been very active in the food industry, in the pharmaceutical industry. Now, what fermentation involves are the sources. So, you have your fungi, you have your bacteria, your algae, or you can use your plant cells, and you provide it an optimized environment to grow. So, you provide it with nutrients that is required, and then you provide it with the temperature, the pH that is required, and you let it grow, and once it has grown, it will produce a product that you want. Now, if I may ask you a very simple question, do you remember the sigmoid curve? Do you know what a sigmoid curve is? Right, it's an S. Now, this is mostly your growth with respect to time, correct? So, you have your lag phase, the log phase, the stationary phase, and that is what we call as a sigmoid curve. Please correct me if I am wrong. Now, for this, you will be mostly aware of fungi, bacteria being used, and that is what we generally do even in the labs. When we are doing a sigmoid curve analysis, you will be using a bacteria or a fungi or a yeast, because they have a very quick growth phase, and that is the reason why we use them for acellular production, because they have a very quick growth phase. They will double in the matter of minutes or hours. If you're looking at stem cells, for example, to make cultured meat, stem cells double once in 24 hours. So, one stem cell will get doubled into 2. So, the growth phase is slow. It's there, but it's slow. So, that's why the production differs for both the technologies. Acellular production is, that's why it is very commonly utilized in the food industry, because you can produce quickly and you can produce in bulk. And then what is involved in the fermentation is your purification of the final product. Another question for you, out of all these steps, growth, development of the product and harvest, which one do you think requires the most capital? Capital means money. The downstream part or the upstream part? You all have had courses in fermentation, I think. So, downstream processing or the purification aspect is the most costly process in the fermentation technology, and that is where the maximum amount of capital is invested, right? Now, fermentation is a very important process. Why? This is the slide that is going to tell you, because it can be used to produce enzymes that can improve protein functionalization or coagulation. One of the example would be production of tofu. Another would be it can be used to develop flavoring ingredients. Now, this flavoring ingredients for Impossible Burger. Have you heard about Impossible Burger? Or have you heard about Beyond Burger or Beyond Meat? Beyond Meat and Impossible Burger are meat alternatives. So, they make plant-based meat products which would resemble a meat product, but they are not meat products, they are plant-based. So, to get the same texture, the same taste, the mouth feel, you need to develop ingredients through fermentation, and that was what they found is in regular meat, you will have hemoglobin. They found soy hemoglobin. Impossible Burger has a patent for that, is soy hemoglobin. So, they found hemoglobin in soy protein or in the soybean, and they grow that using acellular fermentation process or acellular product development, and then use it into their product to provide you that mouth feel, the texture, the feel, the smell, the taste of a burger, of meat, because that hemoglobin adds to that, right? That adds the flavor. It is also used to develop growth factors. Now, these growth factors would be your hormones, the proteins which are required for the cells to grow. For stem cells, you need certain growth factors when you are making cultured meat, and you will learn that they are the most important part when we are looking into the cultured meat production. They are also used to develop gelatin and some of the products like the keratin, the silk. And that is why the fermentation is very, very important in the food industry. Otherwise, people have given you examples of beverages. Yes, beverages, alcoholic beverages, fermentation is there, but that has been there for centuries, right? We all are aware of wine making, beer making processes, but nowadays fermentation has got a very different application from the food industry perspective, and precision fermentation or acellular fermentation or acellular product development is playing a very important role. From the Indian context, you will see as we get into the market landscape, you will realize that in India itself, if you want to set up a startup, the best startup would be acellular product development. If you are able to develop proteins through acellular fermentation or precision fermentation, you are going to help reduce the population of undernourished people, because that is the most important thing, because accessibility to good food, accessibility to nutritious food, that is the most important thing that is happening, and fermentation is playing a very key role in that. Now, another example would be this classic cutlet. I found this as an example and I was very interested to bring it here. Can you guess what it is made of? Is it like does it look like a chicken? Does it look like potato patty? What does it look like? Just guess. There is no marks associated with this. I couldn't find a bigger picture than that, so that's why I don't have it. But this one is a fungi. It's a fungus. The company Emergy Foods. So this is the example of where the whole biomass or the cell is being used as a product. So they grow this fungi, and that particular fungi itself is the product, and they are using this to develop alternatives to bacon, alternatives to chicken. So this is one is the example of classic cutlet, and then you have a crispy cutlet, which would be like a crispy protein. You can find an alternative to KFC protein that you buy, right? And if you look into on the right hand side with respect to the nutritional profile, sodium is there, for example, for the taste perspective. Vitamin D, the calcium, the iron, they are still pretty good in terms of the nutritional profile. But what they don't have is that cholesterol, the fat, the bad fat, which might impact you. And that's why it is also gaining a lot of interest. Other companies that are involved in this area of product development would be At Last Food Corporation and Corn. So Corn, if I remember correctly, is a European company. I might be wrong. Please don't sue me. Okay, I will take my words back. It's a food company somewhere in the world. Now, other examples of fermentation would be development of high source value ingredients, such as the gelatin. Now, gelatin, which is produced through fermentation process, is being used to make gummy bears. Have you heard about gummy bears? It's a very common candy which is loved by kids. Even I do like it. It is also used to make proteins, animal-free proteins. For the milk protein, it is also being used to make different types of milk proteins. So you can get milk alternatives, dairy-free milk. Novozyme is a company which actually uses that to make enzymes. And you can see from the right hand side that it is being used to make food enzymes. It is also being used in the cellular agriculture products. It is being used to make purified proteins, and depending upon the different things. Now, formation or development of these products require a lot of input, and most of the input that we are interested in is the feedstock or the medium that is used for the growth of the microorganisms. We will be discussing that in the precision fermentation. Now, what are the advantages of fermentation? So, it's low cost, definitely, as compared to other unit operations in the food industry. Fermentation is widely used to produce industrial chemicals, feed ingredients, fuels, food products, and others. It's a proven technology. It has been there for centuries. We are aware of how to use it. We know its positives or the advantages and disadvantages, and it's easier to modify. And fermentation processes currently are working at scales of 600,000 liters, and they are being used, and downstream processes as well are very well established for most of the fermentation processes. Rapid production, that is where we were talking about. So, bacteria and the yeast and the fungi, they have a very rapid growth phase. So, they double very quickly. So, you are able to produce more and produce in bulk very, very quickly. So, productivity is very high due to short doubling time. The batch processes only requires a few hours, and it has a very established R&D process. You will see in every institution, majority of the institutions that have food engineering in India or food engineering elsewhere in the world will have someone or at least 2 or 3 faculty members working in the areas of fermentation. It's a very well established and very research oriented program, and it's a very established R&D. Now, let us come into the cultured meat aspect. So, we have talked about the acellular agriculture. Now, let us talk about the cellular agriculture from the cultured meat aspect. Now, the cells are being grown as a product themselves. Now, one of the example that I gave you was the fungi that is being used as a classic cutlet or a crispy cutlet as an alternative to bacon. Now, in this particular case, you are basically using tissues from the animals. So, just like a biopsy, you take out the tissues, you grow them in the lab, you find the ones which are growing well, you take that, you put it into the fermentation process, make it large, make it bigger, and then use that as a product itself. This particular paper is really, really interesting. I would really suggest if you get an opportunity to go through this particular paper. Dr. Chaudhary, who is one of the authors here, was my undergraduate classmate. He's in Singapore right now. I think it is his research group that has published this research article. It's a really interesting research article where they have talked about the cellular agriculture and the cellular production, mostly cultivated meat. Now, this cultivated meat is a genuine animal meat that has very similar sensory and nutritional profile of conventional meat. It comprises of the same cell types arranged to provide the same three-dimensional structure as that of animal muscle or tissue. Okay? Now, what happens is that you take your pluripotent cell sources, the stem cells. Now, these stem cells can differentiate into different types of cells, right. So, it can, some parts of the stem cells will go into muscle formation, some will go into the fat formation, some will go into the fiber formations. So, all of that you separate out, you put that into a fermenter, and once you have put it into a fermenter, you grow it to an extent, you put it into a scaffold. Now, this scaffold is either can be compostable or eatable itself, and you are growing, you provide the nutrients for these cells to grow, and that is where you get the whole structure of the meat itself. So, if you want to make a chicken breast, you take the stem cells, you put it there, you grow it. Now, some of the cells will make the fibers, the muscles, the fats, and then you arrange all of them into like a mold and let them grow. You provide them enough nutrients, and once you let them grow, that is how you will get your chicken breast, the whole design of the chicken breast. So, these are where the scaffolds are coming into picture. Now, I have given an example of this pluripotent cells. So, you are looking at embryonic stem cells, and you are looking at induced pluripotent cells, iPSCs and ESCs, and how they are used. Any questions till now? Go ahead.
Skeletal muscle cell.
No, the scaffold is not basically stating that. What the scaffold is doing is just providing the support for the cells to grow.
Okay.
The stem cells themselves have an idea what they want to become. So, some of the stem cells, because of the DNA that they have, they will become, they will move into providing the fat cells, they will move into providing the muscle cells. So, they will differentiate by themselves. So, once you have identified which cell is moving into where, you collect them. So, it's a very different process. So, you take the cells out of the animal, you grow it in the lab first, differentiate it into different forms of cells that you need. So, let us say if I have to make a chicken breast, as I said, so I need muscles, I need fat, right. So, the cells that are growing into muscles are, I will collect those, I will put them into the fermenter. The cells that are growing into fat, I will take those, I will put them into the fermenter, provide the nutrients for them to harvest, like to grow as much as possible, so I can get enough, and then I take that and I put it inside the scaffold, and then I let them, and I will provide them the nutrients that are required for them to now grow into tissues, like with the muscles tissue, the fats. That is what the scaffold is allowing them to do. That is it. It is just giving them the three-dimensional structure, nothing else.
Okay, sir.
Now, you have understood a little bit of the cellular agriculture or cultured meat. Now, there are areas that are very much of interest and opportunity that exist in this area are the culture media that is being used for the cells to grow, the cell line that we basically use for making the cultured meat, the scaffoldings which are being developed. I have already talked about the cell lines, the tissue culture. All of these areas will provide you opportunities for research. For example, let us say, now access to cell line is a major issue. It is like when we are making the genetic database for the seeds. So, we will go for the wild varieties for different types of seeds. Some seeds will have drought resistance, some seeds will have salt tolerance, some seeds will have another properties. So, we have to collect them, right. So, when we are trying to develop a specific product, we need to have the perfect cell line for that. Access to those cell lines is an important area of research, or development of those cell lines or identifications of those cell lines is an important area of research. scaffolds which are providing the support for your cellular agriculture or the cultured meat, the design of that is very, very important. If you can make it biocompostable, if you can make it biodegradable, or if you can even make it eatable. If I can design a scaffold from starch, let's say, that would be really good, right? So you have the starch, that starch can be consumed, but it's also, what would be the most important property of that scaffold? It has to provide a rigid environment, strength, right? So it should not break when the cells are growing, and it should also be able to allow the nutrients to flow through for the cells to grow. So keeping those properties in mind, if you are able to develop scaffold materials, now this becomes a material science related research, right? But it is completely a biological engineering research. So if you, even if you have a background in mechanical engineering, specifically in material science, with an interest in biological materials, you can work in this area. And that is where exactly what I work on. So we are trying to develop starch-based scaffolds. Now those starch that we obtain are obtained from waste sources. Then you have a lot of research going on in the tissue engineering aspect, making the process much more faster, making the process much more cleaner. And then the culture media. Now, a very basic question for you. Now, if you want to grow a bacteria, what would be the nutrient that you will provide the bacteria to grow? What is the food for the bacteria to grow? Sugar, carbohydrates. What else? Proteins, nitrates, nitrogen, anything else? You keep on saying things, I will say yes to it. Then I'm just asking. The basic, the most important that you would have heard about, okay, fermentation of beer. What is the source? What is the feedstock? Barley, right? Barley is used. What does barley provide? Glucose. That gets fermented into ethanol. Yeast is used, right? Yeast is the host that will change glucose into ethanol. Am I correct? Now, if you are growing cultured meat, what are the things that you will provide for the cells to grow? What is required by you to grow? Do you need hormones to grow? Right? Do you need growth factors to grow? So you have to provide all of that to those stem cells and the muscle tissues and the cells to grow in the scaffolds, right? And those would be mostly obtained from pharmaceutical industries of highest purities, and it's very, very difficult to obtain them. But acellular production, that means recombinant DNA technology, can be used to produce growth factors. That's where your precision fermentation again plays a very important role in cultivated meat. So you are developing your growth factors, or you're producing your growth factors using precision fermentation. So that's another area of research that you have. Oh, sorry, let me go back. So scaffold materials, it has to be sustainable methods of production of these. For the bioreactor and processing, the mode of production understanding, it will allow for the scaled production. The cell culture medium is the main cost driver. The reason being, when you're looking at those growth factors, if you're looking at the purest form of growth factors, they will cost you millions and millions of dollars. I will show you a table later on in the precision fermentation, or I think in this lecture itself. Now let's look into the industry or the market landscape for cellular agriculture. It's actually a growing area, it's a growing field, and India is also there. I think if you are able to see it properly, it would be clear meat or clean meat, something like that. And then what majority of the companies that you are seeing in this particular graph are startups. Now, these startups are also funded by the bigger institutions like Cargill and the bigger players, the Nestle. They are funding these startup companies to get into the alternative meat and alternative proteins. And the global demand for the plant-based meat is also increasing slowly. And that is what this particular graph is suggesting here. Now, most of the companies, what you are seeing here is that it's on a rise. The number of companies that are increasing in this area of research or in this area of production of food products is increasing day by day or year by year. So, 2019 was a very good year for plant-based meats. From the Canadian perspective, Tim Hortons is the company which is provided, like it's a coffee place, like you have CCD or you just call it cafe. You call it Cafe Coffee Day or Coffee Day now? Coffee Day? Okay. So, similar to Coffee Day, we have Tim Hortons in Canada. Now, Tim Hortons at one point of time they started providing us with Beyond Burger. That was a alternative meat, plant-based protein products. So, they went into collaboration with various of these alternative meat protein companies. Burger King has the very similar collaboration. The research from A.T. Kearney, they took into account saying some of the assumptions and what is going on in the market, what is the people's notion, how they feel about meat and what opportunities are there for the alternative meat. What they were suggesting is that by 2040, you're looking at almost reduction of 50% in conventional meat production. So, people's reliance on alternative meat would be very, very high by 2050. So, people will be moving away from chicken, from meat, from beef, mutton and other products, and they will be getting into this cultured meat area. It's an assumed projection taking into consideration what the emotion is right now of the market, but it's I will not ask you to take it as is. Understand the market, understand if you are interested in setting up a startup for this, you have to be 100% sure that that particular product will have a market here. If I come and say I want to set up a cultured meat industry in India, I have to rethink it multiple times, right? I have to find the location. It's all about the location, right? So, that is also a very important aspect in terms of this. The industry, this is another graph that is showing some of the companies that are going with business to business and business to consumer startups that have come up, and how the number of companies have improved or increased from 2015 all the way to 2019. So, the number of companies involved in this cellular agriculture area has increased tremendously, and it's still increasing. More and more startups are coming into this picture. Now, let us look into the other challenges and opportunities that we have. Now, what we are going to discuss here is technology specific challenges and technology agnostic value chains. Now, when you are looking at technology specific value chain, you are looking at the cell line development for the cultured meat. We already discussed about that. The cell culture media development, where you are looking at precision fermentation for growing growth factors. Scaffoldings, developing new types of scaffolds for the cultured meat, and designing new bioreactors, improving the bioreactor optimization processes. From the fermentation perspective, you are looking at the host strain development and engineering. If you are doing any genetic modification to your organism to produce more and more products, the feedstock that you basically use. So, if barley is used for production of beer, is there an alternative that I can use, like one of the food waste that is available, can that be used as a carbon source? If I am able to use a food waste as a carbon source for my fermentation process, I am going to kill two birds with one arrow, right? I am going to reduce the GHG that is being produced because of the food waste, and I am also going to develop a product that is going to add monetary value. Purification from the host, downstream processing is another area of research which people love to work in. I work in that area. developing new types of downstream processing techniques which will give you the highly purified products is very, very important, and development of new bioreactors or designing new bioreactors. Now, when you get into plant-based meat alternatives, you're looking at crop characterization optimization, raw material optimization again, and the production methods that are basically used. And if you're looking at the technology agnostic value chain, that means those that are not related to technology aspect, not engineering, let's say more on the management side. So, you're looking at the financing and services, the business services, the end product and reproduct lines that you have to come. So, if you are working in the food science or developing new products, this would be the area of research that would be very, very interesting. Like, what is the current demand of the market, like what people are interested in. If there is, for example, when we are talking about the genetically modified organisms, right, the GMOs. Now, GMOs are something that we eat on a day-to-day basis. You would be amazed to know that there are countries in the world that have banned GMOs for anything to do with baby food. You cannot have any form of GMOs for baby food. And that's a good thing, in my view, as a parent, I will say, if I have a notion in that aspect. But there are certain rules and regulations, right? So, if you want to get into those type of research, you have to understand the policies of different countries and how these products can be launched there. Now, Good Food Institute, for example, from where I have taken this particular graph and some of the other sources, the sources that I have used for my presentation is the Good Food Institute. Now, Good Food Institute has been lobbying for alternative meat for a very long time, and they are a big supporter of that. And I like the idea, and I really appreciate if you can go onto the website of Good Food Institute, and you will find amazing resources on cellular agriculture, on fermentation, specifically for precision fermentation, and how this alternative meat or alternative or plant-based protein industry is basically increasing, and what are the demands for it. So, they have done life cycle analysis, they have done techno-economic analysis onto different products, and they have identified what works, what doesn't work, and if it has to work, when will it work, whether it will work in 2024, it is going to work in 2020 or 2050. They have identified what is happening, the trend of majority of these areas. So, brokerages and distribution, you are looking at the safety, QA and regulatories. Not every country allows. So, if you are looking at precision fermentation and making cultured meat is not approved by various countries, is still at very approval stages. The FDA in USA has not approved many of the company's products yet. They are still looking at what are the what do you say, the challenges associated with it, what might happen if you are drinking it. Now, these regulations change from country to country. I will give you a very basic example of it. If you buy the real juice here, the orange juice, let's say from the real juice company, that's made by Dabur, if I understand correctly. Now, on their Tetra Pak, do they say rich source of vitamin C? Do they say that? Yes or no? Or if you drink this, it will cure cancer. Can they say that? You will see, and you'll be amazed. If you go to different countries in the world, some of those countries will have this type of promotion on their Tetra Paks, that this can cure this, this can cure that. Why cannot they say it? Do you know why? Or just guess. Why cannot a company say that this particular juice, or if you drink real juice or real orange juice, you will be curing cancer? Why cannot they say that? I'm so sorry, I was not able to hear you. No, there are proven evidences that it works, but Why cannot they say that?
Hello. Recently in FSSI in India, they said like no products in India should come with any health claims, and they have given a apart from that they said like this health claim cannot be given in such a way that it won't give the exact result whenever any products, for example, there are a lot of products is added with fortified like fortification is also done. Apart from that, there will be these products contain a lot of bioactives and all, but it won't give the exact result for all the community or all the people. It will vary with respect to persons.
So let's say, if I remember correctly, there is a medicine called as Dolo 650, which if you have fever, you basically have that. It's a paracetamol.
Yes. Yes.
Now, it's called 650 because it's 650 milligrams. It's a dosage. So if I take one tablet, it will reduce my fever. It's all about the dosage. Like how much of that orange juice do I need to drink to get cancer free? Can somebody tell me that? No, they cannot, right? And that's the only reason why they cannot mention that. Because you were right, like they cannot make that health claim because you don't know like whether you need to drink 10 packs or 4 packs. It might so happen that you might need 2000 packs. For somebody it will work in 2 packs, but we don't know that. So that efficacy is not available, and that is the reason why these things don't show up. And that is also the reason that people have some concerns. Like for example, if I remember correctly, there was a case against Bonvita for claiming certain things in India, right? The same thing happens with the cultured meat products. Like when the moment the ingredients that are being used, are they safe? How safe are they? We don't know. So a lot of research is going on in that aspect also. So that's also another opportunity for students to work in if you're looking into research areas for this. The QA and the quality assurance and the safety of the product that is developed through precision fermentation or the cultured meat products, that's another area of research. Now, other opportunity would be into the fermentation side. So you're looking at a novel strain engineering, so identifying new strains, using the right feedstock. So as I said, like if I can use a food waste as a carbon source, I might be able to reduce the total production cost. If I can reduce the production cost, I can reduce the cost of the product. Then identifying or enhancing alternative protein products, so providing high value ingredients from plant-based meat or cultivated meats, and high volume, low cost manufacturing. So scale up and low cost manufacturing is another area or opportunity that exists for us. My research goes into this, valorized waste streams, where we try to use different types of food waste materials, agricultural byproducts that can be used or valorized into mediums that can be used for the cells to grow. Now, this example, so this particular slide is talking about the medium, the cellular medium or the culture medium that is basically used. So the biggest cost for cellular agriculture comes from the cell culture medium, and it is often estimated with the 80% of the marginal cost of the end product. Now, FBS or fetal bovine serum or the serums, these are used for the cells to grow. These require growth factors. They have the growth factors and other things in them. So if you can develop a serum free system, that might add or that might help reduce the overall cost. So serum is highly variable product, and any variation. So you don't get the same serum is not like if I buy it from this brand and I buy it from another brand, they are not the same. They'll vary. So you don't get the consistency in growth. So you need consistency. Now, the batch which is produced this year might not be the same as the next year. And the serum such as FBS is the main ingredient in cell culture media, as it contains hormones, lipids, and growth factors that are essential for the cell proliferation. If you're the opportunity that exists is in developing or using serum-free media. Now, where the advantages that it provides is consistency. So, because I'm not, how do you produce these serums? Do you have any idea? Not the serum-free media, the previous one. How do you produce FBS? From animals, right? Like live animals or dead animals? Live animals, right? Or dead animals? Live, dead. Live. So, it is produced in live animals, you extract, right? So, the point is that it will not be consistent then. It can never be consistent. So, that is why you need a serum-free media. So, the advantage is the consistency, the standardization, the safety. You can control the safety. You do not know. There are many things that you might not be able to control elsewhere, but you would be able to control. If you, okay, it is just like you developing something and you have control about on every aspect of it. If you have control on every aspect of that development of the product, you would make sure that the safety is considered, the quality is considered, the consistency is considered. So, better control over the cellular functions, ease of purification, enhances cell growth, viability, and expansion, and ease of formulation. So, disadvantage would be the cost, and the cost, because serum-free medias are costlier than FBS, because more control you have, more you spend. Because every ingredient that you use, think about it. If I am using the FGF and the TGF, these are your growth factors. If you look at on the right hand side, how many zeros are there? That itself is approximately $8 million. The amount per 20,000 liters is 2 grams. For 2 grams, 4 million. So, the level of purity that you have to have is pharmaceutical grade. Pharmaceutical grade purity is very, very high. Food grade purity is lower. If I am able to use a food grade level of purity for production of cultivated meats or for cellular agriculture, I would be able to reduce the cost of production. That is another aspect to work on. And it is all because of the growth factors, that all the cost that is involved here. So, this was this particular work was done by Good Food Institute, where they identified the total cost of producing, and they also identified how you can reduce it. So, if you are able to use food waste as the source for growing as a feedstock in the fermentation process, for growing these growth factors using precision fermentation, you would be able to reduce the cost of production for these growth factors. So, working in those areas is very, very important. So, what are the key takeaways from this particular set of slides on cellular agriculture is, the cellular agriculture can unlock opportunities to develop next generation meat over and make it more sustainable. Continued growth of alternative meat or alternative protein will provide the required push for the cellular agriculture industries. This field requires multidisciplinary knowledge to accelerate or sustain the current progress in the plant-based and the cultivated meat sector. So, you all have an amazing opportunity because of the multidisciplinary background that you have right now, and there is a substantial room for innovation. So, this field is not going anywhere. You have a lot of time, you can do a lot of research in this area, and you will be seeing in the next coming 5 years or 10 years, there is so much of research that has gone into, and there will be products that would be available in the market at that time. Okay? So after this particular slide, we are going to take a break for 15 minutes and then we will resume again on the precision fermentation. Thank you. Yeah, we can definitely answer the questions from the first slide.
Can you comment on the cost of these alternatives? Do you think it is comparable to the ones which is like previously existing, or because what we saw, it feels like it's more, way more, because you started your slide with affordability. And based on the Indian market, the scenario doesn't look very promising in India.
From the price point of view, you're saying, right?
Price point of view.
Okay, to answer your question, let me give you a bit of a story back. Okay. So when I was born and brought up here in India. So majority of the food that we get was grown by small scale farmers on their farm next to our house, and we buy them directly from them, right? And I would have never seen them using any chemicals in their fields. So technically, if I call that, I can call it like organic, right? If there were no chemicals used to produce the food. So I ate organic food for a very long time. When I went abroad and I went to the shop to buy, let's say, to do groceries, to buy some fruits and vegetables, organic food is sometimes 2 times costlier than the regular fruits and vegetables. Similar way, an alternative protein, until unless the cost of production can be reduced, its cost at present time would be higher than what is available conventionally. It's all about the production cost, right? And that is where we were talking about if we are able to produce the growth factors using feedstocks which are cheaper or which are even free, we might be able to reduce the total cost. So I will seriously ask you to go and look into the life cycle analysis conducted by Good Food Institute on alternative proteins. In that, they have explained different scenarios in which if you grow these growth factors with feedstocks from here, you might be able to reduce this. If you use these type of cell cultures or cell lines or hosts, you might be able to reduce your cost by certain percentages. But they did reduce it by, let's say, to around $4 from millions. So that is possible, right? And it will also depend upon different it's hard to answer that question completely, but it will depend on the, like, from the production perspective. If you are able to reduce the production cost, you can reduce the cost, and you can make it more affordable. but it will also depend upon how people are interested in it. Like in India, there was an article in which they were talking about that 70% of people in urban areas make really poor choices in terms of the protein they eat. And I think it was in Times of India or some news article was there. I will try to find that. I will share it with you all later on. But that suggests that even if the affordability is there, people can make poor choices. So even if, let's say, this particular product is affordable, people might not still want it because of different notions that they might have, that I don't like GMOs, so I might not go there. Right? So there is that aspect. Any other question?
Yes, sir. So like again, based on the Indian scenario, we have a lot of unskilled laborers as compared to skilled laborers, and precision fermentation will require more of skilled laborers and automation. So how to bridge that gap? Because just poultry breeding, that is comparatively easier a process for an unskilled labor to earn money from as compared to learning these, getting that education and doing like performing it in a real life scenario.
It's actually a very good question. I was never expecting that type of question actually. See, fermentation can be divided into different stages, right? The procurement of the feedstock is one stage of the fermentation process, or development of the feedstock for the feed material. That might not require a lot of skill. So that can be where you can use the agricultural, so the paralis that are being burnt in Punjab and Haryana, that feedstock can be used. So you might not need a lot of skilled labor for that. You can still use your unskilled labor there. So if you are able to develop a whole industry that is supporting the fermentation process, you might be able to provide employment to that unskilled labor. Then comes the downstream processing, which requires a highly skilled individual. The fermentation will require semi-skilled individual because everything is controlled. And with the advent of AI, things are changing. You might not even require people to control many things, and there will be robotics or the sensors are controlling everything. But you need a highly skilled individual to be looking after all of that. But I still think the feedstock production side, the initial part itself, has a lot of opportunities for the unskilled labor. There is a possibility. You have to look at it from a different perspective. That's why I said, like, there is that opportunity exists in the technology aspect and the agnostic part of where you don't need the technology. So how do you market it? How do you implement it? And you have to convince the people, you have to convince the governments that you can use this. So I am trying to convince you, saying that, hey, let's use that paralis to make the feedstock. So you will get interested because that's a waste for you. You're not making any money out of it, and you would like to make money out of it. So that sort of conversation is required to get into that. We'll discuss more on that, on this precision fermentation also.
Okay, sir.
If you have any other question from the previous? No? Okay. So welcome back for the second round of lecture. So this is where we are going to discuss about the precision fermentation. So I gave you a bit of background on precision fermentation. We did discuss some part of it as we were discussing the cellular agriculture aspect. Now, precision fermentation comes from this particular term itself, precise. It has to be very, very specific to a product. Now, how do you make fermentation very specific to a product? You don't want anything else, but you just want the product that you're interested in. Is by making sure the host or the yeast or the fungi or the bacteria or the prokaryote or eukaryote that you're using in your fermentation process is only able to make your product of interest. That's what precision fermentation means, or precise means in this aspect. Again, as a part of the outline, I'll give you some background in the fermentation. We did have this conversation already, so I might go a bit quickly, or if in case I can explain a bit more in that area. and then how the precision fermentation works, how it is done, what are the targets and the host selection. Now, what sort of host is required to grow what type of product is very, very important. The feedstock selection, we have been having that conversation as part of the question that you asked, and also the fermenter design, optimization, and the scale up. And then we will talk about the application and the challenges and the industry landscape or the market landscape, how it looks like for precision fermentation. Now, fermentation based production, as we have already discussed, requires sources which are your microorganisms like fungi, bacteria, algae, and the plant cells. You need to optimize the cells for the fermentation by optimizing the cell culture medium. Now, the question for you would be, so if you know that yeast will grow if I give a specific feedstock to it, right? So, if I give you yeast and I am giving you the feed for it, so will you take this and put it in the fermenter directly, or will you start it at a lower scale, like in a lab first? What will be the process? Lab scale? Why lab scale? You have to activate it? Oh, you are activating the yeast, okay. I give you activated yeast. You have to acclimatize your yeast to the production conditions, to the pH, to the temperature, right? And then you want to see whether the product is growing, or sorry, your yeast is growing enough and it is developing the product or producing the product, right? So, that is done at the lab scale. So, you do the culture. Once you have enough culture, you use that as a starter culture and you put that in the fermenter and provide all the nutrients for it to grow, let it grow, optimize the fermentation condition for it to grow, and then you have your product developed, and that is what you use. So, acclimatization of the yeast to that particular raw material that you are providing is very, very important. The yeast can produce a product from different sources. So, let us say it is just a question, because all of you have a background in biotechnology, majority of you have a background in biotechnology, and you would have had a course in this. So, let us say if I am using a carbon source 1 and then I have a carbon source 2, will the yeast fail? Which source will it use first, C1 or C2? Why? Just because I am writing it as a C1? Yeah, but then how did you make a choice that it will use C1? I did not say that C1 is the more likable by the yeast. So, it will always use the carbon source that it likes. So, it can be C2 also, right? That is why I said why C1. So, it can be either of the carbon sources. When it has finished one carbon source, it will start using the second carbon source, but then it has to make modifications to its internal machinery, right? Its whole biochemical pathway has to change to accommodate that particular carbon source to grow. Am I correct? So, because if you start eating one specific food product right now, okay, this was the best example. If you go to southern India, you eat idli sambar there, and if you go to northern India, you eat idli sambar there. They are two different idli sambars, right? If you are going to south, you cannot expect the same taste and the texture that you get in the north. If you go from the south to the north, you cannot expect the same texture and the taste. You have to get used to it, and that is the reason that acclimatization is required for any fermentation process. The type of carbon source that you are providing and the consistency of that carbon source has to be maintained for it to produce your product in a specific way. Am I correct in that? You are able to understand that aspect, right? So, that is where the optimization aspect comes into for your cell culture growth or the growth of your host, which is going to produce a product. Then comes the fermentation, where your cells are added to the bioreactor with the culture media, and then they proliferate. So, basically you have already acclimatized it, then you take that, put it into the fermentation. The fermenter is just used to grow it. and make it big. Increase the number. So, first lab scale, you have to do it at a small scale. You can use a 2-liter fermenter or a 5-liter fermenter or a 10-liter fermenter, and then you move on to a bigger, larger scale fermenter. Now, when you are doing this scale up, that requires a lot of mathematical calculations. We will be discussing that later, and that is the reason the whole course that you will see, you have the bioreactor design analysis, which will be coming in the later days, where you will learn how to design a bioreactor. What are the equations, what is the mass balance required, the heat balance required to maintain a bioreactor or design the bioreactor for a specific product. And then comes the purification and the final product, or the proteins are isolated and purified. That is the downstream processing, right. Now, they are used as food ingredients, or the products that comes out, they can be used as a food ingredient, they can be used as pharmaceutical products, right. They can be used as food beverages, that becomes a food ingredient too. So, what we have understood is fermentation is very, very important, but it's a long process, not from the time perspective, but it requires different stages. That is what I mean by the long process. It has multiple stages. Now, we have already discussed this slide in the previous session that we had. So, fermentation is key for the food industry because it can allow you to produce enzymes that can be used to add textures, color, or any profile, a sensory profile to your product. Like tofu, it can be used to develop recombinant products like soy hemoglobin, which were used by Impossible Burger to make their Impossible meat or Impossible Burger, which was a meat alternative. They are also used for the growth factors for fermentation. Now, this is where I would really be interested in some of you in future can work in this area and use feedstocks, specifically the waste products or the food waste that we are developing, we generate every day. You know how much of the food waste is generated in the agriculture sector in India? How much of our agriculture produce is wasted in percentage? Guess. 10%, 20%, 30%, 1%, 2%, 30 to 40, 40. It depends. If it is somewhere between 40 to 50%. No, you are much more unique. You are giving me exact. Yeah, so it depends. It's 40 to 50%. It all depends on the cycle, the whole storage facilities that you have, the logistical facilities that you have, because you are going to generate waste at every stage of the food supply chain. Now, if all that waste can be used as a feedstock for your fermentation process, because they still contain the nutrients that can be used by the microbes to grow. So, if you can use those as your feedstock, you would be able to reduce the overall cost of production. And you can, if you are able to use those to grow your growth factors, suddenly your production cost from $4 million will reduce to 200,000 or even less. Now, we have already gone through this application of the MRG foods, the classic cutlet. They also had crispy cutlet. I would really like you to go onto their website, themeaty.com, and look at the products that they have. If you have a phone or an iPad or a tablet right now, sorry, or even a computer, you should go onto their website and look at it. The products that they develop is really amazing, and it's really interesting how they do it. Now, that is where the whole cell biomass is basically used. So, because we did discuss that in the fermentation process, in the acellular, either it will be the product, the cells are producing only the products, or the cell itself is the whole product for us. They are also acting as a source of high value ingredients. We discussed about the dairy proteins, the enzymes, and the gelatin. We discussed about the advantages and disadvantages, not disadvantages, but mostly the advantages of the fermentation process. You will see there's more or less a repetition of some of the slides from the previous one, because I had to bring back you to that discussion. That's the reason why I brought these the same slides, some of them here. So it's also because of the low cost, the proven technology and scalability, the rapid production, the established R&D. Now there are a couple of things that we can definitely discuss here for these. Now let's talk about the low cost aspect of fermentation process. Why we call it a low cost process? Why it is a low cost process? No, you need. You're saying that it's not energy intensive, right? No, like if you're doing precision fermentation, it is actually very energy intensive. That's one disadvantage of precision fermentation.
Because the microbes can be grown on a variety of livestock, which like we don't need to be dependent on just synthetic media, which is costlier. You can go for alternatives like waste. So if we have a feedstock which has a carbon source and a nitrogen source, we can just grow the organism and we can get a product out of it.
Okay, so that can be one. Any other? Just read the slide. What do you understand from there? Rapid production is there, but why the low cost? The low cost part I am talking about, the fermentation is widely used in to produce industrial chemicals, feed ingredients, fuels, food products and others. So if there is a technology that is used by multiple industries, so if I am developing a product that I can sell to multiple consumers, will I increase the price or reduce the price? I have so many buyers. Right. So that's why, because it is widely used, you have multiple players that are available in the market to make you the technology. So that's the reason why fermentation can be, but there are not many companies actually that actually make. Sorry, there are not many companies in the world that actually make reactors. It's just, I also say, I think this because of the scale at which you can use them. You can develop more bulk products, you can reducing the cost of it. It's a very proven technology, scalability is there, that can also be there of reduced cost. Now let's talk about precision fermentation. Now fermentation, as we all discussed, is a very old process, a biotechnology method which has been used in food production. During the introduction, Dr. Sabyasachi did tell you about breweries and beverages, the alcoholic beverages and non-alcoholic beverages that are being developed or produced using fermentation process. It is basically or traditionally has been used to enhance the nutritional and the functional characteristics of food, improve the overall food sensory properties or food safety. So we improve the food sensory properties by producing the enzymes that can be used. So fermentation has been used to make those enzymes. Now precision fermentation is just an advanced version of the regular traditional fermentation process that we have. If you remember, we did talk about precision fermentation. I told you that the recombinant DNA technology was used to make it. So that was what the precision fermentation used to be. And then we started naming it as a precision fermentation. So because you can use recombinant DNA technology, so you're using molecular biology, making modifications to the host, the DNA, you're adding your own DNA of interest, you can develop products, you can make your host develop products that are of your interest. So you can make different types of products, you can make proteins, you can make vitamins, you can make antioxidants. Rennet is a very good example. Rennet is used for where? Cheese. And rennet used to be produced from? It's very hard to hear, that's why I'm saying.
Calf intestines.
Okay. So initially, yes, but if you can do it through fermentation, You can make it consistently, right? It will have a consistent efficacy, and it is widely utilized everywhere. But that is also one of the product, and it is also one of the product which has been developed through this recombinant DNA technology that has been approved by everyone. Every country in the world actually allows use of rennet. Human drugs like insulin is produced using recombinant DNA technology. Now, let's look into different types of the fermentations that are used in the food industry. So, you have your traditional fermentation, you have your precision fermentation here, and then you have your biomass growth, just for the growth of the biomass. So, microbes used under anaerobic conditions convert sugar into end products with unique textures or flavors. So, what you are using is you use traditional high value raw materials to produce end products. Example would be milk and produce yogurt. So, end products minimally or not processed. So, the end product is not processed. Yogurt, bread, tempeh and alcoholic beverages are part of your traditional fermentation process. When you are looking at the biomass, that is where the mycoprotein comes into picture. So, biomass itself serves as an ingredient. It uses novel inexpensive feedstocks to produce high value proteins. Example, agriculture remains. So, that is where your paralis can be used. So, instead of like burning it, you can utilize that as a feedstock for growing some of the in the fermentation process to develop mycoproteins. And then comes the precision fermentation, where the host usually is not present in the final product. So, the final product is developed by the host, but the host is not the final product. Sometimes yes, but not all the time. It uses novel inexpensive feedstocks. We can use again the byproducts to produce the proteins. High purity, you require very high purity in that. If you are developing insulin for human consumption, let us say for human usage, it has to be very, very of high purity, right? It has to be clean. So, for that your downstream processing capital cost would be very, very high. So, understanding this, which one would be costlier, fermentation or precision fermentation? So, precision fermentation, yes, fermentation is advantageous, yes, we can grow in bulk, but it is a highly processed product. The product that comes out from precision fermentation is highly processed. So, you are adding cost to it. So, this high cost, this low cost. I love this particular technology because it can utilize any bioresource that you have, any feedstock that you can provide. Even your food waste can be used to grow mycoproteins. This is something that we all should be looking at, honestly. With the current air quality indexes that you are seeing in different parts of this country, this can be one good example or one good unit operation that can be used. Now, how is the precision fermentation done? In precision fermentation, we use a host, and that particular host is called the factory, because that particular host is the one which is producing your product. And that host DNA is modified by introduction of a foreign DNA, the recombinant DNA technology is utilized. And what you are doing for that genome modification or genome editing, you can use this Cas9, which is basically going to cut the genome DNA at different locations. You can insert your your target DNA there in that particular location where the genome of the host has been cut. So, you have your target selection. So, upstream part will be your target selection, what you want to make. So, proteins, lipids, pigments, vitamins. Then comes the host selection or the strain development. The host is selected for target expression and strain functionality is optimized. Question here for you all, can all act as host? Like all the bacteria and the fungi and the yeast can act as host? For let us say if I have a product A, or like a specific protein that I would like to make, can I use E. coli for it, or should I be using yeast for it? Should I be using a fungi for it? How do I decide which host I should use? What will be the criteria for the host selection? Ease of extraction, you are talking about downstream processing part. Yes, definitely. What else? I am still not able to hear you. Sorry.
Substrate for fermentation. Growth media.
The growth media?
The substrate for fermentation.
Substrate accumulation.
For fermentation, like.
Okay. Okay. What else?
The target product should be the part of its metabolism. If it's not produced,
If you do genetic modification, you can actually make it a part of its metabolism. That is what we are going to discuss tomorrow in synthetic biology. That's another, that would be systems biology. If you understand the biochemical factory, which goes inside a microbe, you would be able to twist it, turn it in such a way that you would be able to produce the product of your interest. Yes, but why? How? What would be the other, like, general requirement for host selection?
It should not be pathogenic organisms.
Okay, it should not be pathogenic.
It should not produce any metabolites which will react with the
product. So it should not produce anything else other than your product, let's say. You should be able to produce your products first of all, and in good amount. Downstream processing, ease of extraction has to be there. What else? Oh, so you're talking about the moisture content part?
Yes. Like we are going to use fungi. We basically prefer a solid substrate based fermentation, where substrate is comparatively less that we use for a microbe like bacteria.
See, he actually brought up a very important aspect. Depending on the host, you will define what type of reactor you will use.
Yes.
Right? So everything is related in this aspect of fermentation process. Right? Many a times you might be use solid state fermentation process. You can use submerged fermentation process. Now, which one is cheaper, solid state fermentation or submerged?
Submerged fermentation.
Okay, think about it again. Solid state fermentation is cheaper or submerged fermentation is cheaper? I made a face and then you are starting saying that. If you say submerged fermentation, you have to answer why. If you say solid state fermentation, you have to answer why.
Sir, submerged fermentation is scalable. We can scale it according to as per the at what scale we production want.
Okay.
But solid fermentation, we are like there are a lot of constraints regarding size, how to increase, manage, controllability. So might be submerged.
Okay. From the energy perspective, which one is cheaper? So if I'm looking from the energy, see, that's the thing. Depending on the question that I will ask, the answer will change. Like if you're looking for a production capability perspective, yes, submerged will be able to give you more. You can develop more products in that. It's scalable at a much higher rate than a solid state fermentation system. But from the energy perspective, solid state fermentation is cheaper as compared to submerged state. But that would be defined depending upon the host that you're using, or depending on the feedstock that you're using, right? Depending on the feedstock, the type of fermenter that you will use will change. There are different things that are interrelated here. Similarly, again, let's get into the feedstock selection. So you have to have the sugar or the industry side streams that can be used as our feedstock in the fermentation process. So these are done before you get into the fermentation. Then you're coming into the fermenter design. You have your submerged, the solid state, or the membrane cell recycled bioreactors. Now, when do we use membrane cell recycled bioreactors? When you are working with a very high value product. Then you come to the process optimization, the pH, the temperature, the oxygen or no oxygen concentration of the salt. Which of these fermentation systems between the submerged and the solid state, you would be able to have a much better control over the fermentation processes, like the pH, the temperature in the submerged, right? So again, depending on if your product or the host is not dependent on the pH, or if it is not dependent upon the temperature of the oxygen, not too much of variability in that, you might be able to select the type of fermentation system. Either you go for submerged or you will go for your solid state, right? And then comes the downstream processing. You might require some pretreatment, you might require separation, concentration, purification, product formulation. All of this adds to the cost, which is always going to increase. More and more steps are added, much more would be the cost. So now think again, in precision fermentation, we are making a very specific product, right, of a very high value. It's a very high value product, that means we need it of highest purity. If we need it for highest purity, our downstream processing would be more. So the cost of the product would be more. So it is a very nice technology, but it's also very expensive when we are looking at precision fermentation. This is my personal view. I'm not saying that this is a general view, but this is my personal view. My understanding about precision fermentation is that it's a costly process. Now let's look into the each and every aspect of the precision fermentation. That would be the target and the host selection. So the precision fermentation process starts with the selection of the target product. So you have to know what you want to make first, and only then you can decide whether you want to go through the precision fermentation or traditional fermentation process. So this is followed by the selection of the host that would be able to express the desired product. We already discussed almost all of this in the previous slide, but selection is also based on cost effectiveness. Which one would be cheaper for you? Now, ease of downstream processing was one of the answers, like easier extraction of the product. Now, host selection also requires a thorough understanding of the different metabolic pathways or for the strain development. Now, that was an amazing answer. Whoever gave that, thank you for that. If you understand the metabolic pathway, you can make changes to the host metabolic pathway to develop the product of your interest in more quantity. So new strains can use inexpensive feedstock. Now, if you make modification to the host biochemical pathway in such a way that instead of using sugar, it can actually develop an enzyme that can break cellulose or something else to get the sugar that can be used later on for the production of your product, you're reducing the cost. You're making a new strain, but you can use a different type of feedstock for your process. And then you can have an improved fermentation efficiency, that means shorter cycles and higher yields, and you can also have simplified downstream processing and better product recovery. Now, I'll give you a very, let me ask you a very simple question. What was like you have heard about corn being used to make bioethanol, right? And there was this food versus fuel. This is a classic example that is given to everyone in the world. Whoever comes to the undergraduate or graduate life, you will hear about this food versus fuel. What was it? So when the corn was being used to make bioethanol, everybody wanted, because you will get more money out of it, people started growing corn for bioethanol production. So the cost of corn for human consumption, because availability decreased, the cost increased. It's a demand and supply thing, right? So that's the food versus fuel part in basic, like in the most layman way I can explain. But what is the biggest challenge? Was the efficiency of that corn being used for bioethanol high? The conversion efficiency, was it high? It was not. Why? Why do you think the efficiency of conversion of the lignocellulosic biomass, that is the corn stalks things, was less?
Will make more time.
Okay.
time
What's
is
the
one.
biggest difference in plant cell and animal cell?
Actually, I am not that much. I am from mechanical background.
That is fine. Okay,
So
what is like if I give you steel and if I give you wood?
wood can break easily when
And
compared
then you
to
have a
this.
steel. So that's the biggest difference between plant cell and animal cell. Again, which one will break easy? Okay, biotechnology guys.
Animal.
It's easier to break the animal cells because you don't have a. In plant cells, you have a cell wall, and it's not easy to break them. It's not easy to digest them. So people started making modifications to the corn genome so that they can grow corn with walls or cell walls that can break easily when it comes into the mechanical process. That was one of the research, if I remember correctly, was being done by one of my friend long time back when I finished my undergrad. So think it from that perspective, like you have to do modification to the growth of the plant itself to get a feedstock. What if you can make the modification to the feedstock? Now that's a costly process. So if you can develop an enzyme that can actually interact and can break down the cell wall, break down and make it more available, or the resources, make it more bioavailable for your host to grow, then yes, it is possible. So that is where genetic modification of the host itself sometimes is required to improve the overall productivity. C'est bon? When I say c'est bon, that's in French, it's like good? Correct? So you have to do genetic modification. So that is another important parameter when we are doing the host selection process here. Now you can look in from the table here. So for the bacteria, the prokaryotes are easier to manage. The cons are genetic drift, higher mutation rates. Now bacteria will mutate very quickly, and it has a very bad public perception. So if you say that, oh, I am producing something from E. coli, people might not like it. So there are pathogenic E. coli, there are non-pathogenic E. coli strains that we use. Again, when you're looking at the yeast and the molds, it's slower generation times, which is a con for it. It could require more nutrients, and sometimes potential mold are pathogenic. Mycotoxins production is one of the issues that is there, and it has a negative impact on the product rheology. If you're using fungi, the rheology will change because fungi itself will add the texture to your product. Then it comes to the microalgae. You have target extraction challenges, which are presence of tough cell walls, which is silica based coatings, and autotrophically grown, some like slow growth and low density. Oh, feedstock selection. The choice of feedstock is one of the major contributors to the overall cost. So we have already discussed this in detail for the process efficiency and the process sustainability. So as we go into the detail of the precision fermentation, the sustainability aspect of this whole course, this is where the sustainability part will come into picture, because we are trying to develop, or we are trying to choose feedstocks that are sustainable in nature. How do you define sustainability in one way or other? Give me the most layman definition of sustainability. So it is nice for the environment. How then what is sustainability? Break the word. Sustain and the ability to sustain. What is sustain? To maintain, like, see, it's not about to retain just the future. It's about you have a sustainable source to it, you have sustainable access to it, and it's always there, right? So you have the availability of that feedstock at all times in the same quality, in the same consistency is maintained, and its impact on the environment is also sustainable. It's good, but it's also sustainable, it's maintainable, right? So that's where the sustainability is not just from the environmental aspect. Think about the accessibility and availability aspect to it. So accessibility, availability of the feedstock should be there. So that's where the food waste becomes a major source. It is an amazing source that can be used, but do you think food waste is a sustainable source? Just give me an answer, yes or no. So, if you say yes, that means you are okay producing more waste. If you say no, then how is it sustainable? Every process has waste, that those waste can be used. I am not talking about the waste that we generate, it's the whole process that generates. If that is something which will always be existing, we can use those waste as a feedstock. That is what the intent is, right? So, then you have the conventional feedstocks, which are your standardized conditions. So, no food safety issues, but it is costly to use, less sustainable, and example would be refined sugar, which is used for the fermentation process. Alternative feedstocks, not standardized. So, that means our food waste is not a standardized waste or a feedstock that can be used, it would be different. So, the batch that I might be getting from Punjab of all the let us say the lignocellulosic biomass will differ than the batch that I will get from Odisha, right. So, I will not say it is standardized, it is a sustainable resource, but it is not standardized. There comes pre-treatments, that is why there is a little bit of cost which will get impacted there, because we might have to do some pre-treatment to make it consistent and standardized before we use it. Production inconsistency comes because it is non-standardized, and then regulatory challenges, food safety, because I do not know if I am using food waste. So, let us say if I go to the landfill area, I start taking all the waste from there to make some food products, will you agree? No, because there are food safety concerns. You cannot use that as a food waste, sorry, food waste as a resource for making your food products. So, safety concerns have also been there. Then examples would be the side streams from the food industry, the lignocellulosic plant materials which are used in the fermentation. So, leafy woody parts of the plants remains after harvest, used for mycoprotein production. So, that is where I was saying the paralis can be used for the mycoprotein production. The byproducts, food industry, the apple peel from the juice processing, that can be used for alternative proteins. Now, apple peels, the citrus peels, do you think we will be using that in the submerged fermentation or solid state? Apple peels, you have the apples, you have peeled the apple peels, potato peels, onion leaf, the peels, will you use it in submerged fermentation or solid state fermentation? Easier would be solid state. So, depending upon it, you can change it. Like, see, I have used onion peels for solid state fermentation for natural color. It is a cheaper process, but it is a longer process, but it is a cheaper process. Plastic waste, conversion of plastic to edible proteins by microbials after pyrolysis steps. So, potential for various other applications that can be there. Agriculture crops, plant rich in carbohydrates such as sugarcane can be used for alternative protein production. So, these are some of the examples that you have. Any questions till now?
Sir, you stated that we can use the paralis. So, what is the commercial viability of paralis? Because there is major issue in the state, that is the collection issue and the processing issue also. And as you stated, the variability issue, that the variation might occur in the paralis which are generated from Punjab and Haryana and other states. So, is there any research or ongoing research on the commercial viability of those paralis?
See, I cannot give you example on parali itself because I do not work here, but on agriculture byproducts, yes. So, think about this way, if I may, because we are deviating a little bit from the topic, but it is an important question that you have asked, because when we are using those as a feedstock, let us say for the fermentation process, we would like to maintain a consistency in the quality of the feedstock that we are getting. Semo?
Yes, sir.
So, what happens? Okay, so think about this. So, this is one field, a couple of fields that you have. What is the biggest challenge for the farmers? They are saying that they don't have the resources to collect it.
Yeah, balers and etc.
Right?
Yes, sir. Time also, sir, because after the harvest and before sowing, there is only 15 to 25 days.
Yeah, so now you think about in such a way. Now, let's say if the commercial importance of that lignocellulosic biomass that is spread out on the field is very, very high. Do you think a company will pop up saying that, you know what, I will come and collect it? You just tell me the day that you have harvested, I will happily come. I will come even at the night and collect it. Do you think that will happen? If the commercial value of that particular feedstock is very high.
Yes, sir.
It will happen, right? So, we have to first develop that commercial value. The only way you can do that is through research, where you can show that these lignocellulosic biomass are commercially viable and can be used to make products of very high value. So, when you make products of very high value from a low value waste, the commercial value of that waste just went very, very high, and importance became very high, right. So, that is what is needed. That is my personal point of view.
Yes, sir.
That is what is needed for people to understand that these lignocellulosic biomass have a lot of commercial value. If it becomes, let's say, if you are able to establish a pre-processing facility in a specific district in Punjab, and it has collection facility that people from there will go to all these farms and collect it and bring it to that pre-processing facility. Now, that pre-processing facility is basically collecting the products or the lignocellulosic biomass from all of these farms in one of the district, bring it to that particular pre-processing facility, and that facility is processing and making a consistent feedstock. Now, that inconsistency has been fixed by consistency. What do I mean by it has been fixed? I will give you a very basic example. The orange juice that we were talking about, the real juice, the orange juice that you drink, do you think that orange juice that we get, if I take a batch of oranges from a farm, squeeze the juice out of it, will it have the same vitamin C, the sugar, the acidity that I will get, that I would have got last year, or that I will get next year? No, it will not, right? Industries don't work in that way. They collect all of that juice in a vat. Do you think this juice is pasteurized? It is pasteurized. Yes, so, at what temperature? At a very high temperature. I will not ask you to give me the exact value of the temperature, but it's higher, right? Yes, sir. Higher than 60, 70 degrees Celsius, or even 80 degrees Celsius. Vitamin C degrades at 40 degrees Celsius. So, all the juice that you have just pasteurized, will it have any vitamin C left? So, how do they make it consistent? They have to add it. So, the food industry adds vitamin C after the fact. The aroma of the orange juice that you get, the smell of the orange juice that you get, the moment you open, you get an orangey flavor, right? You have just heated everything. Lemon has a fragrance, right? So, lemon, which you squeeze on a salad, it gives you that aroma, the volatiles that comes out, it gives you that smell. If I heat it, will I still have that smell? So, when I have pasteurized the orange juice, will I still have the smell? Technically, no. How do you add the smell? You add it. You don't add a smell gas, but you add the fragrance. Who makes that fragrance? Give me one fragrance company name. Don't give me Fog, other ones. Have you heard about Chanel?
Yes, sir.
Right?
Yes.
Coco Chanel?
Yes.
Chanel makes fragrances for food industry. The smell that we get in our juices, in many of our food products, is actually made by Chanel. It's actually costly. So, what you eat contains a lot of this. So, that is adding consistency. So, in the end, the product that is going to the consumer is consistent. You are getting my point where I am taking you. I gave you that example to explain that you get the feedstock from different sources, you bring it, you process it, you pre-process it, you process it in such a way that you get a consistent feedstock, and then that particular industry will sell it to the bigger industry that, hey, here is the feedstock that you can use. So, now you have added value. So, this industry is making money from the bigger industry, the farmers are making money from that collection industry. Just you add the value. Okay?
Yes, sir.
That was the question, right? Okay, let's get into the fermenter design. Sometimes we are deviating away from it, but it's a very interesting conversation. Thank you. Now, fermenter design, optimization, and scale up. So, the type of bioreactor which is selected is based on their ability to meet the specific requirements of the process or the product that you are trying to grow or the product that you are trying to obtain. So, the type of bioreactor you are going to use will change. It also depends on the target products, the host that you are going to use, what type of host, what are the conditions that it requires for its growth, whether you are going to use submerged or the solid state fermentation system. So, but then that is where you will see that you have the lab scale where the screening is done. Then you go into the bigger lab scale. So, you go 2 liters or 5 liter fermentation system. So, in-house process simulation and optimization. This is where majority of our research goes. So, if you come to my research group, a colleague of mine, we bought like 2 bioreactors of 5 liters each. So, we do this type of study, the lab scale bioreactor design, bioreactor application, and then we get into the pilot scale, and that pilot scale can be scaled up into the industrial scale value. So, optimization of the process parameters such as the temperature, the pH, the oxygen supply, or sometimes you might not require an oxygen. So, you might require a completely anaerobic condition. So, this all affects the overall cost and sustainability of the process. Okay? Okay, which one is cheaper, anaerobic fermentation or aerobic fermentation? It's a very genuine question. It's a very simple question. Which one do you think is cheaper, aerobic or anaerobic? Aerobic? How many of you are saying aerobic? Only 3? Yeah, I can say only 3, 4. How many of you are saying anaerobic? Again, similar numbers. You are equal in numbers. Why? Okay, if anaerobic, why? If aerobic, why? Okay. Sparging air, there is a separate need. Oxygen. So, do you add oxygen through air or do you add oxygen as an oxygen? Is costly or cheap? That adds cost, right? So, aerobic processes are costlier as compared to anaerobic processes. That is where only the cost factor comes in, nothing else will change. Aerobic processes are a bit costlier than anaerobic processes. Now, some of the applications and the challenges with respect to the fermentation would be some of the challenges that need to be overcome when we are looking at precision fermentation specifically would be the new strain development and the sustainable feedstock, because if you can develop strains that can consume feedstocks that are waste materials, that would be amazing. You are going to reduce the overall cost. The process scalability with the large investments and overcoming public perception is key. Precision fermentation requires recombinant DNA. Right? You are basically genetically modifying your organism. So, the product that you are producing is coming out from a genetically modified organism. So, public perception is I do not use GMOs. So, your product will not have any traction in the market. So, you are basically trying to, you have to convince the public that the product that you are developing is good. Right? That notion has to be overcome. So, new fermenter designs are required to meet the needs, and the regulations. We need to convince the regulatory authorities on product quality and safety. Now, that's where the FDA, the EU, Food Safety, FSSAI in India will come into picture if they allow GMOs or if they don't allow GMOs into the market. And if they do, what are the criteria? You have to meet all those standards, all those criteria. So, these are examples of some of the products, the status quo with what is happening with them. So, for the dairy, it meets the need for the animal-free milk, proteins with the same composition, taste, benefits as animal ones by using more production, sustainable with the most advanced and skilled way. It's a very cost-effective, demanding, but demanding on a purification process, because the moment you are looking at making dairy-free milk, you're looking at producing protein, and those protein has to be processed. The downstream processing, you need highly purified proteins to make that product. So, your downstream processing cost will go up. Similarly, other examples are there for the fats, the oils, and the eggs. Then comes the regulatory authorities part. So, where you are looking at the EU. So, you have the EFSA, and on the US side you have the FDA. So, FDA has a requirement that the ingredients that you are using in your product development, if they are GRAS, that means generally recognized as safe, it will get the approval. EU is way much more stricter as compared to US from the food safety perspective. They don't allow genetic modification. In many of the countries won't even allow you to sell GMOs. So, they are very strict in those nature. So, that's where the market, if you are developing a product that is coming out of a GMO, you don't have a very large market then if the public perception is not changed, or if you are not able to convince these regulatory authorities. Good Food Institute that I named in the previous presentation is one of the lobbying group that actually lobbies in the favor of alternative protein industries, the precision fermentation industries, and the cultivated meat industries to these regulatory authorities, trying to convince them that it is safer from the product quality perspective, and it's also safer from the environment perspective. Now, there are also changes, there will also be certain regulations with respect to the labeling process, which would be involved, whether you are using GMOs, non-GMOs. Some countries will allow you to write that you have to mention GMOs. Like in India, you have those green dots, red dots. If it is vegetarian, you have to have the green dots. If it is non-vegetarian, you have to have the red dot. Am I correct? So, those type of regulatory combinations have to be used. Now, the industry and the market landscape, you have already seen one of the slides where the meat, alternative meat or the plant-based meat substitute requirement or the market is increasing. And there are also a lot of companies that have come into picture with respect to the precision fermentation. Now, the market is expected to grow from 1.6 billion, which was in 2022, to almost 36 billion in 2030. So, it's a market that is still growing, and precision fermentation is being used for alternative meat, so alternative protein generation or protein production. So, it's a growing market. So, if you are interested to look into for startups in this industry, this is the time. The time is ripe for you to get into that. Now, the few of the key takeaways from this whole presentation today would be the precision fermentation process is a technology that is rapidly being adopted by food tech companies. There is a continued growth of alternative proteins and the meat industry, and it will provide the required push for the precision fermentation. The technology enables food producers to efficiently create specific proteins, lipids, vitamins, enzymes, pigments, and more using microbes instead of animals. You have substantial room for innovation. The key challenges that still pertains with respect to precision fermentation would be the strain development, the host selection, the strain development, alternative feedstock for sustainability, process scalability, proper infrastructure, consumer education, and regulation. So, if you are able to, or if you are able to at least fix 40 or 50% of all of these challenges, you would be able to have a very huge market for this. I hope you have understood or you have got some idea about precision fermentation and cellular agriculture. I know many people, I can look at the faces, you are all tired and hungry. So, thank you. That would be it for today.